Thank you for visiting our poster at the Great Lakes Bioinformatics Conference. Today I'll be discussing our project, Dogpile, and the various pipelines for processing and analyzing whole genome sequencing data. Next generation sequencing technologies aided by computational tools and access to state-of-the-art high-performance computing systems have revolutionized the field of genetics, ushering in today's genomics era. The decreased costs and improved technologies have enabled researchers to rapidly generate whole genome sequence data of individual samples of interest and even entire cohorts, yielding massive amounts of data. These large-scale sequencing efforts have been especially fruitful in veterinary medicine and in particular dogs. However, processing and analyzing these sometimes immense data sets can be a major undertaking unto itself that is not always straightforward, can be relatively non-standard, and is prone to potential reproducibility pitfalls. To address these hurdles, we've developed a series of pipelines comprising the current best practices and publicly available resources from the Broad and GATK, particular for processing whole genome sequences, and we've packaged them into this. These pipelines include single sample genotyping, joint genotyping, novel variant discovery, and allele frequency analysis. Both the single sample and joint genotyping pipelines are written in the workflow management system, SnakeMake, to ensure both reproducibility as well as enabling scalability. First, the single sample genotyping pipeline in figure 2 here processes raw sequencing files through the generation of what are genomic variant call format files, which are basically a complete description of the entire sample genome. By providing a simple file of sample IDs to process, all the required pipeline input is generated, which includes parsing sequencing files for various metadata items and is sent to a specified directory for submission to, in our case, a HPC system here on campus, the Minnesota Supercomputing Institute at the University of Minnesota. And figure 2 then depicts these various GATK-based steps that a single sample goes through from user input to execution via the SnakeMake pipeline. And to process a single sample in a reasonable amount of time, so like 24 to 36 hours, over 400 cores spread across nearly 150 individual job submissions are ultimately required. The second pipeline, the joint genotype pipeline in figure 4, leverages population-wide information in the form of GVCFs generated by the single sample pipeline to joint call short variants. And by joint calling variants, we can achieve substantial gains in overall variant calling accuracy. This would be increased sensitivity for identifying low frequency variants. This is the ability to rescue variants from sites with low coverage. Figure 4 illustrates the overall joint calling pipeline and input strategy, where instead of storing large amounts of data in primary storage, we are able to pull down our stored GVCFs that are part of the cohort and then joint genotype from there. Joint genotyping can be a computationally demanding endeavor, and to minimize these costs, we developed a strategy to break the genome into manageable intervals or regions depicted in figure 3 and process each of these smaller intervals in parallel. And this is at 300x in figure 4. This is the total number of intervals we broke the canine genome into. Our most recent pipeline, the novel variant discovery, enables the identification of novel variants that are private to an individual sample. So these are mutations not observed in the joint variant call set. This pipeline has been decidedly successful in identifying likely causal variants potentially linked to, as of today, a handful of canine monogenic disorders. There's also a potential for clinical use, which is really exciting. We see these rare inherited disorders in puppies, for example. We may be able to figure out what's going on genetically and make informed decisions for either treatment or for a breeder. Finally, our allele frequency analysis pipeline calculates per site allele frequencies by group, so cases and controls, enabling the identification of variants associated with various Mendelian diseases and traits in dogs. This has also been highly successful with a number of published papers from ourselves and some of our collaborators. In conclusion, the pipelines included here in this Dogpile can genotype single sample, whole genome sequencing data, joint genotype entire cohorts and identify novel variants, as well as calculate allele frequencies for groups. This was designed specifically to lower the barrier to entry while encouraging reproducibility and analyzing whole genome sequence data that is often faced by researchers with perhaps the institutional resources like Minnesota Supercomputer Institute, Computing Institute, but lacking the bioinformatic acumen. We, the Canine Genetics Group in the Department of Veterinary Clinical Sciences, have processed whole genome sequence data for over 600 dogs across more than 50 breeds. And in collaboration with various research groups have successfully employed these pipelines to aid in the disentangling of the genetic basis of various disorders and traits in our canine companions and ultimately humans. Finally, although developed for canine researchers, our pipelines can be modified with relatively little bioinformatic know-how to work effectively in other veterinary species. And we'd like to thank, finally, we'd like to thank MSI, the Minnesota Supercomputing Institute, for providing all the necessary resources and support that have enabled this project. And we also want to thank our collaborators at various institutions for providing some of the sequencing data as part of our population. Thank you.